Now, when you were started training in genetics in the 1970s, what would have been...
Did you have to say that? Oh! And what attracted me was you could have a change in a single gene that had a profound impact on the biology of the organism. Right. And so, in contrast to biochemistry, which is everything's changing all the time, nothing's static. Biochemistry, it's sort of the what's going on in the cell at any one moment. Okay?
Right.
And so we still have a very imperfect knowledge of that. It's like trying to get an idea of the city of Montreal by taking a picture from the top of the city and looking at all the traffic moving and people walking and trying to figure out what's happening. That's what biochemistry is.
Right.
Whereas in genetics, if you change one gene, you might get a profound abnormality, a child who's supremely smart or is disabled. Right. Or whatever. So I liked the absolute nature of it.
2009, the Toronto Star hailed your research on the protein NETO1.
Yes.
It was a collaborative effort at Sick Kids.
That's correct.
Now, was this a change in direction? I mean, you've been doing eye genetics. This is now synaptic genetics.
You must have to do a lot of work to do these interviews.
He did have no idea.
I'm sorry. You know what you do in research is you go where the research takes you. So, right. Throughout my career, I went from studying inherited biochemical diseases and then diseases of the retina, and then the reason we got into the area of the particular gene and protein you mentioned, NETO1, and there's NETO2, is because I had a brilliant young graduate, probably my most talented graduate student ever, started in my lab around 2000, David Ng. And we'd been working on a particular class of genes for about 10 years in my lab. The class of genes I alluded to earlier that was similar to the one in flies, and frankly it was getting sort of boring. And so I said to David, why don't you go look for a gene that's important for development, that's only found in the eye, and that is conserved in biology, and that nobody else is working on. So he did a computer analysis of such genes. There were about 13,000 of them in the gene database at the time. And he found one out of the 13,000 that fit the criteria he and I had set out. And so the database indicated that this was an eye-specific gene, but as soon as he started to study it, it was clear it was actually more expressed in the rest of the brain than in the eye. But by that point, it was clear it was so interesting, I wasn't going to let it go. Yeah, it was related to other genes that were important for development and that were located in synapses, and it was novel and nobody was studying it. So David worked on this project. In fact, for about 7 years. PhDs take a long time, and he actually stayed. He met his wife, who was another graduate student in my lab, and so he stayed for an extra 2 years doing a postdoctoral fellowship. And so this gene we discovered, NETO1, is a protein found in synapses, and it helps one of the major proteins that's involved in sending signals between nerves. It facilitates the function of that protein. And when
it's involved in learning
and memory. Yeah.
Yes.
And when we got rid of this protein called NETO1, the learning and memory functions were impaired in a very subtle way.
Okay.
So that was nice. But the cool thing was we found we could compensate with a drug for that learning defect. So it was one of the first examples of showing you could correct an inherited learning defect with a drug. I think it's kind of a wonderful achievement that this has been discovered, because I think we all thought that if you have an inherited learning defect, you're set in stone and you don't know.
Yes, exactly.
And this shows that isn't necessarily the case.
Yes.
And so I think the marriage of genetics and neurobiology is going to bring this to medical reality and hope, gradually, and it won't be able to help everybody. It'll be able to help some individuals for sure in the future.
This is very exciting.
But there's another part of the story I want to finish.
Yes,
so I mentioned that David Ng met his wife in my lab. So then he's now at Columbia University training with one of the world's leading neuroscientists named Tom Jessel. But anyway, they got married, and they came to my door. They were leaving for New York in January of some years, about 3 or 4 years ago. And in their hands, they had a baby and 2 bound PhD theses. So that's the best output I've ever had from my lab. You know, you don't earn millions of dollars studying genetics. Well, some people do, but I don't. But it's I guess what's attractive is the feeling that you can contribute to the body of eternal knowledge in this way. That's pretty cool.